So let's take our seats. We have a very interesting topic. Big biotech, where is it going? But big biotech will also involve small biotech and all of biotech. And I have a remarkably distinguished set of panelists who've had a whole host of experiences, which I will not try to repeat. They're all in the material that you got when you came to the meeting. But to my immediate left is Jane GROGAN, and she's Executive Vice President and Head of Research at Biogen. And next to her is Pushkal Gar. And to get this correct, he's the Chief Medical Officer and Executive Vice President in Development of Medical Affairs at Alnylam. And next to him is Ray DESHAIES. Ray is the Senior Vice President, Global Research Emeritus at Amgen. And next to him is George YANCOPOULOS. I hope I got that right. Close. Good shot. Board co-chair, president, and chief scientific officer of Regeneron. So a lot of experience in the room, all the way from new biotech, only 20 years old, and older biotech, and middle biotech. And I think we'll have a very interesting conversation. Now, I will call on a couple of people with a question, and they will banter back and forth or talk, and then we'll open for dialogue between panelists. And at the end, I'm hoping to have time to invite questions from the room, but it'll depend on time. So let's start thinking back. Genentech was established in 1976 with the aspirations and then fulfillment of becoming a vertically integrated worldwide marketing biotech company quite successfully. 50 years later, the world has changed. There's a lot of large biotech, mid-sized biotech, small biotech. So if you think about innovative new ideas, is the age of the vertically integrated aspiration in biotech history? What's the models for bringing innovation into this community and industry that new idea, a new aspiration appears, and what are the models one should think about, or do you disagree that it can still be the aspiration of a small company or an entrepreneur to develop a vertically integrated, fully functional biotech? So, George, do you want to comment on that? Because you've lived through most of that.
Well, certainly Genentech was our dream and our role model. And I think the whole concept of building a fully integrated, company from beginning to end can solve for a lot of the needs and the problems. But the reason why we aspire to be like Genentech really did not have to do with all the downstream stuff. It had to do with the fact that at the time, they were the leaders in innovation. And that's, I think, what distinguishes success. You need a lot of the downstream capabilities. I think there's other ways to actually handle the downstream capabilities. I actually believe in some cases they could be done, better using collaborations. We actually built our company based on 50-50 collaborations. We've got Chris Wiebacher here from Sanofi. That relationship provided a lot of that downstream vertical integration stuff And without that, we could not have taken things all the way. But it all starts with the innovation. That's what's really hard. That's what's really limiting. So I always think whether it's a project that we're working on or something that we're trying to do with the company, you got to understand what's the hardest thing, what's the true bottleneck, what's really the limitation to success. And it starts with innovation. And that's what's most limiting in the industry. This is the hardest industry to truly innovate in. I mean, innovating an iPhone, that's not innovation. iPhone 16, 17, who the hell cares? What we do is true innovation, and it's quantifiably the hardest thing that human beings try to do. And that's where I think the focus needs to be. The downstream stuff can be useful to have, it can be important to have, it can maximize the value that you create. And so I'm not minimizing, it's incredibly important. But the innovation is where the focus should be, and what we as a society should be figuring out how to much more greatly incentivize for and try to promote through society, let alone in the biotech industry.
Jane?
Is this on? Yes. First off, thank you for having me here in this panel. And as we think about Genentech, we have to think about Biogen, and that's not just because I'm currently head of research there, but sitting next to the founder of that company, which is still the independent pharma established in, 1978, I believe, in the East Coast, that is still existing. So that's quite some accolades. And I know at the time that was also being aimed at building a fully integrated organization. One of the spirits of that company, and I think this gets back to what George was saying, was really driving after innovation and high unmet medical need, and really taking on the challenges of breakthrough medications in really novel and new areas. And that's certainly something we continue to pioneer, I think, as everyone does on this panel as well. Innovation is key. I think you're going to hear that again and again and again at this panel. I think one thing that's changed in the last 4 decades is that not one organization or not one large pharma can actually drive all the innovation within the walls of its company. And you need to look outside. And so the ecosystem is always used and partnered with academic sites where those innovations often happen. But I think we're shifting a few decades later to much more of an equitable ecosystem where who's doing what, how do we integrate with them, how do we work together on ideas so we can grow together in industry. And there are times when there's efficiencies by bringing things in-house because you can streamline things. There are certain platforms that you need to bring in-house. But otherwise, I think a lot of things can be really accessed outside. And I really like the idea of that, 50-50 or even 60-40. How far can we really build efficiencies and then build agile organizations to move our industry forward together?
I want to challenge you because innovation is important, but if you're starting a small firm, you got to have resources, meaning money, and you have to have people. People are really a lot of people trained in this industry now, but the ability to finance a company through A and B round, and then to get resources to develop products that are really innovative takes time. And for Alnylam, which is sitting next to you, which I happen to have a lot of time, it took 10 years. And it took about $1 billion. So are those days, that challenge is really difficult right now.
Yeah, I mean, I can speak a little bit, and thank you also, Phil, and to the Galien Foundation for inviting us to speak. I think, again, getting back to the innovation piece, I think the question is there a better alternative to biotech? And I think, we haven't talked enough about sort of the innovation culture, right? And the culture is the critical part. What we do, as George was saying, is incredibly, incredibly hard. It should be hard, actually, to attract capital to ideas, right? It shouldn't be sort of easy. It should be hard because we're trying to do hard things. And there's a lot of people with interesting ideas. It's not $1 billion all at once, right? It's incremental based on a tenacious team that's able to generate interesting data and do the right experiments to show the value of what they're doing. And I think sort of the model of having, a real proximity between decision makers and the science, having clear accountability on who's making those choices, risk-taking, I think that's a very unique aspect that really fosters that innovative environment. So I think that is the best way. I think, you know, as we, I second, you know, support wholeheartedly the whole point about, you know, partnerships. I mean, Phil, you've been part of that. We've all, you know, with Sanofi, with Regeneron, we've done with the medicines company. So we've built the company based on that. And there's been places where we can actually go it the whole way as we've gotten larger to bring, you know, medicines directly to patients. And there's places where that hasn't made sense for us. And we've done partnerships to do that. So I think now when we think about really some other areas, which is when we think about neglected diseases and rare disease, that's where I think we really do have to think about different models, because I don't know that the current biotech model necessarily supports that as well.
Ray, do you have any comments?
Yeah, you know, there's an interesting point in there you made about the time span of technology development. And I would argue it's even a little bit longer than the number you put on it. So if you look at when a technology first starts being developed in industry to when you have products that are approaching blockbuster status, you're typically in a window of about 15 to 20 years. The first monoclonal antibody, J&J, Mironimab, approved I think in '86. It wasn't until Rituxan crossed the blockbuster threshold in 2002. So that was 16 years for antibodies. Antibody drug conjugates, the first one approved again was in 2000, and it wasn't until the mid-2010s that Kadcyla crossed that barrier. So again, in that 15 to 20 year time range. And you see the same thing with Alnylam. So, you know, the problem is in industry, if you look at people who are leading R&D or people who are leading research, they are not in those jobs for 15 to 20 years. Right? You'll probably have 2, in some cases 3 rounds of turnover. So you're in a very long cycle business with short cycle occupancy of the leadership chairs in that business. And so it's difficult to sustain in a large company that kind of intensity of commitment for that duration of time without a financial return. So I think there's always enormous room in this industry for innovation to be happening in small startup companies that are being financed in other ways. And, you know, part of the problem is biology, you never know how something is going to work or what's going to work. Take genome editing, right? It started with approaches like Sangamo, and then it went on to TALENs, and CRISPR wasn't even discovered yet, right? And so there's so much serendipity in biology that I think you need a it's like evolution, it's like variation selection. That's our industry, variation selection. You have enormous amount of variation created by capital, pension funds, university endowments, funding venture capital. And from that variation, you are going to select the few things that work and then use the muscle of big companies to help drive the clinical trials that you need to do to bring it to market. I just looked the other day for a completely other reason. I'm writing a review, and I looked to see, of the top 10 selling drugs in the world, how many were discovered, how many were invented at the people that are selling them. And I could only find 3, and one of them was dupilumab. And dupilumab, of course, you have a partnership to help you in developing that molecule.
2 of the top 10.
Yeah. Yes.
Invented at Regeneron.
Aflibercept is another one. Yeah. So, you know, I think to me that speaks volumes that, you know, 7 of the top 10 selling drugs in the world were not invented by the people that are selling them.
And I think, if I may, I think that's true in these vertically integrated companies, because the way they got their molecules was not necessarily that they invented them in-house, sorry, George, except for Regeneron, but that there was a mindset around what's the science that you're trying to follow and the question you're trying to answer, and then having that sweet spot of expertise within an organization that could see an opportunity outside with which you could then go and partner with someone or build a molecule together or buy in that asset as well. So that external relationship has always been part of how we've built this, you know, the industry that we're in.
You know, I want to build on a point that Ray said, which I think is incredibly important, about short-term leadership in a long-term business. And it gets back to your point about funding innovation. And I think we should really talk about that to a degree. I mean, we have a real communication problem. I think there should be a lot more incentives, and we're, as a society, talking about actually destroying those incentives right now. But getting back to Ray's point, you know, our story is even longer than Alnylam's. It took us 20 years for our first approval, but then we average 1 approval, including 5 blockbusters, a year, okay, since then, for 15 years. Why? Because we have something that nobody else in the industry has. We've kept our teams together. I'm looking at like Jamie Orengo, who heads our immunology and inflammation group. She looks so young, but she's been 15 to 20 years.
Oh boy,
15
do you
to
know
20
how.
years. And so we all learn together, all of us. We built systems and platforms, and we're using that, and it is reproducible. But the system generally, we can get into, and a lot of it was partnerships, how we survived for 20 years without a product. But what we realize is that the system we now have in society is counterproductive to trying to create the long-term sustainable innovation models that can actually then reproducibly deliver drugs. I mean, I think Alnylam is a great example, but it takes a miracle, it takes a lot of work to just get to that point. And we have to understand the system is broken, and we're doing a terrible job of communicating how important this system is and how we shouldn't be among the top 10 industries, the one that has the worst return overall on investment. I mean, we should be number 1. Why? Because we're addressing the thing that people care about, okay? You know, you care if you lose your iPhone, but if you find out that a loved person has a horrible diagnosis, you care a hell of a lot more, and we're not creating the right system that rewards the right things that we should be rewarding in society.
Okay.
And I think one thing interesting about the Alnylam example is, is that the discovery of N-acetylgalactosamine as being a ligand to get into the liver was absolutely critical for driving success of that platform. But there were major companies that sort of invested in that platform and exited that platform before that discovery was made. And it just took that decade plus to get to the point of making that discovery to really open up the platform.
And a few very good chemists. Okay. What?
We were talking about this a little earlier, chatting over lunch, but you know, there was a comment made earlier on today around there's too many Me Too drugs out there in the industry, but the actual fact is, if something fails, if a medicine fails, it doesn't mean necessarily the target has failed. We've learned that a good old-fashioned antibody targeting a known cytokine, if you do it the right way, it can be transformative. And so the modality matters, and sometimes hanging on and really understanding that the target remains a really important biological target with which to go after, then hanging in there sometimes is really important.
So we've talked about the environment for these innovative companies. If you look forward over the next decade and what's going on in biological science now, what excites you, Pushkal, as to how the industry is going to make a difference?
Yeah, I mean, I don't know if I'm going to have anything terribly profound to say other than I think we'd all agree, we're in a remarkable era where, you know, from 30 years ago or so when the Human Genome Project was sort of underway and people felt, somewhat disappointed that we didn't have sort of an immediate return on that investment. But that's actually paying off now. I mean, the work that, you know, Amgen and Regeneron did in terms of building up large genotype-phenotype databases, those insights are now yielding tremendous scientific advances. And so, I mean, we just heard a lot, you know, earlier about, you know, immunology and targeted therapies, cell therapies, but I think where we are with genetically targeted therapies through a variety of modalities, whether it's gene editing, it's siRNA, it's ASOs, it's messenger RNA, all of these things, I think we're in a very, it's a remarkable thing. We actually have molecular scalpels to basically take out one specific, you know, disease-causing gene or protein and perturb the system in a way that will be beneficial to patients. I think that's just a remarkable thing. It forces us to actually, you know, sharpen our game in terms of understanding biology, causal biology in humans, right? Because you're being that precise. And the other, you know, and now we're going to be able to go into more and more tissues as we work on delivery. We'll hopefully find ways to cross the blood-brain barrier, and there'll be combinatorial approaches now for polygenic diseases, I think, increasingly. And then I think the pharmacology of these different approaches is also fascinating because most of them are turning out that you can actually give them either once or very, very infrequently. And so I think we have the opportunity to be on the precipice of basically like vaccine-like approaches in the next 10 or 15 years for some chronic diseases. And so that's really a remarkable opportunity that I think we have in front of us. If we can really harness all the power and intellect that we have in science to make a big difference. So I think it's going to be really revolutionary over the next 10 or 15 years.
Ray?
All right. I just submitted a review last week to Nature Reviews Drug Discovery, and maybe one of you in the audience is a reviewer, so I'm going to give you a plug. Maybe it'll help my review get accepted. So I am really excited by what I see as a broad advance in our ability to make what I would call multi-specific molecular medicines. So these are drugs that work by contacting two or more things, either simultaneously or sequentially. And where contacting two or more things is fundamental to their mechanism of action. And what we've seen is a major upswing in those kind of drugs. So I just did an analysis of this in 2017, 2018. My first 2 years in industry, there were 105 NMEs approved by the FDA, 3 of which, according to the way I define it, are multi-specifics. In 2022, there were 38 drug NMEs approved. 8 of those were multi-specific. So it goes from less than 3% to more than 20% in 5 years. Right, so massive, massive growth in a very long cycle industry.
The most important number you said in there, how many drugs were approved in 2022?
38 NMEs. Yeah.
Okay, how many biotech companies are there?
A lot.
5,000.
And ongoing clinical trials.
That's the thing that we got to understand. Less than 1 in 100 companies will ever come up with one drug. That's how hard this business is. But go on. But I agree. I'm a fan of multi-specifics, so keep going.
So why am I excited?
It's so hard. 38 by the entire world. I mean, that's crazy.
So why I'm excited about multi-specifics?
Several of those were by Regeneron.
Your market cap isn't there yet.
It's about the future.
Okay. There's so much love between Amgen and Regeneron.
All right, at the science level, there is love. Let's not confuse what happens at different levels.
Yes. So the reason I'm so excited about multi-specifics is, look, I'm a biologist. I'm not really a drug developer. And when I look at drug development, it strikes me that biology throws 3 huge challenges at drug developers. The first is redundancy, right? That the way systems are wired, you can block one pathway. It's like getting from LA to San Francisco. There's so many different ways to do it. You can take a car, you can take a train, you can fly, you can take a boat, you can ride a bicycle, you can walk, many different modalities. Well, circuitry in human cells is very similar. You block one way, and there's another way to get it done. So redundancy is a massive challenge. Second massive challenge, therapeutic index. We reuse the same parts again and again and again in different cells to do similar or different things. Here's an interesting statistic. The number of parts in the 777 Boeing airplane, 132,500 independent, distinct. These are different types of parts, not 10 rivets. That would be one part, okay? 132,500. We have fewer than 25,000 parts encoded in our genome, yet we design and build the 777. So we're obviously more complicated. So we can do that because we use the same parts over and over and over again. Even if you have perfect specificity, you will have on-target toxicity, almost guaranteed. So that's the second challenge. The third challenge is people like me, when I was an academic, discovered genes that would be very impactful in disease, but that are completely undruggable. And so multi-specifics allow you to go after all of those challenges. Redundancy by building in more than one activity into the molecule. Therapeutic index by attaching a zip code to the molecule, like GalNAc for siRNA. And undruggability by making molecules that are matchmakers some of the ones that Regeneron is building, for example, that bring T cells to cancer cells to kill the cancer cell. Well, the drug itself is no longer the pharmacological agent. The pharmacological agent already exists in the body.
So I want to say the following. The major unmet need in all of biomedical science is in neuroscience. We have not made progress on schizophrenia, manic depression, many other problems, Alzheimer's, Parkinson's, modest progress at best, hopefully better. Come on, boys and gals.
Well, I think this gets back to what I was saying before. You know, when I went to college, I was a triple major, physics, math, and chemistry, because I thought these were the hardest sciences, okay? Eventually, I migrated to biology. Why? You know, if you're smart, you can solve physics problems. You can solve math problems. Biology is an unknown. In order to cure diseases, most of the time, you need to understand them, okay? And biology, what Ray was talking about, there's 25,000 genes, probably more encoded in ways we don't even understand. But we don't understand how it all works together. And the diseases that you're talking about are amongst the most complicated diseases where we are so far away from understanding the biology. How are we going to really cure them? I hope genetics will provide insights and tools, and we have approaches with our friends here at Alnylam and others so that we can attack them. But it all comes down to how complicated biology. It's the most complicated of all science. Okay, I think it takes individual genius and dedication and incredible human, you know, thinking and work in order to solve these problems. And how does this get solved? And getting back to what these guys said, it's all about new game-changing technologies that give you abilities to attack things that you couldn't attack before. We got to understand the biology. Neuro, we don't understand most of the biology, so even with the tools, it might take us a while to really, really make a dent there. But it's all about the tools. Like these guys were saying, I mean, in my world, it was, you know, we were pioneers in soluble receptors. That led to a whole class of drugs. That was a big drug class. I mean, somebody had to think, you got to make soluble receptors. It's not, you know, AI can't think of it. A human brain had to think of it. You had to make a human antibody mouse. That changed the world for us. Bispecifics and now multispecifics. Genetic medicines, siRNAs, that's why we're partnering with these. It's all about these technology platforms. Those will ultimately deliver the drugs. But as Ray said, it often takes years, decades, from the initial insights or thoughts or whatever about that technology to leading to a stream of the drugs. So this is why the business is so complicated. There are very few game-changing technologies. In my lifetime of 35 years at one company, you know, running R&D, okay, you know, there's probably less than 10 game-changing across the entire industry, okay? And we're still struggling to really understand biology, and we hope new tools, large-scale genetics, and all this will help us. But it's hard, and it's so dependent on real genius and real innovation.
Can I add to that
No,
too?
I was going to
No?
call
Yeah.
on you if you hadn't.
There we go. We're in sync, Phil. To your point, it's understanding the disease, and that is using new tools. And so I think one thing that was not mentioned was the biomarkers and the advent of tools with which we can probe and understand baseline human disease. And the second part is on treatment. Like once we start to get new treatments for neurological indications, and these are happening, we have our very own with Leqembi and follow-on, you know, tau-lowering agents for Alzheimer's disease, as well as progress in ALS and SMA, for example. But as we start to be able to dissect the data from patients who have failed or not failed on these therapies, we'll start to understand a lot more about what that unmet medical need or just what the basic biology is that's driving those diseases as well. The challenge with the brain is it's hard to measure. It's not like a cancer treatment where, you know, you've got a cancer and it goes away, or you can see it on an image and you can see that in a very short amount of time. You know, we're dealing often with, you know, chronic debilitating diseases that take a while to measure kind of the lack of progression in certain cases. This is different if you're thinking about neuromuscular diseases, but I think therein lies the hope and the challenge.
Okay.
I'd be interested to get your perspective on something I've wondered about, Jane, which is if you could routinely get the specificity to distinguish between closely related subtypes of receptor in the CNS. That's very hard to achieve with a small molecule. Let's say you can wave a wand and get antibodies into the brain at a high concentration. Would that solve a lot of the issues that we currently face with drugs, particularly for neurology?
Yeah, I don't have an absolute answer for that, and I don't think we know. I think the hope is, I think it comes down to biodistribution. We still have to work out where in the brain our drugs go and where they need to go and at what concentration they need to get there. And then we need to have to worry about things like the redundancy issue that you were talking about. So can we saturate the right area of the brain with, our colleagues at Roche are doing this kind of thing with an A-beta lowering antibody linked to a transferrin receptor. Will that be sufficient then to get to the right parts of the brain as well? And then will that carry with it greater or smaller toxicities when you think about that therapeutic index that you were talking about as well?
Yeah, and you're talking about a multitude of cell types in the brain. We talk about the brain, but then we're talking about multitude of cell types and tissues, so it's very.
I mean, when do we need to target microglia, right?
Exactly.
Microglia are really important cells. They phagocytose. They take up all this protein. We want to ramp them up, but maybe when we find out someone has Alzheimer's, that is already ramped up and we need to go to another pathway to target. The field just doesn't know this yet.
There was a paper recently said they transfer mitochondria to neurons. So,
Metabolism is key to everything.
okay, so biology is complicated. One of our take-home message, biology is complicated. One of the great creations of the human brain is computer science, AI. We've even gotten to the stage where we can use it and train it to produce images, text, ideas possible. That means it's not copying, it's relating and making something new. So is AI going to be the solution? How do you view the impact of AI on this industry? Pushkal, since you're an expert in AI.
Yeah, I'm clearly an expert. So all I can say is I have no profound answer other than it will, but I don't think we know how, right? I mean, I think we have to recognize that there's changes afoot. Right now, everyone's focused on, mundane things about how do we kind of just take, create efficiency and reduce white space, et cetera, and that's all well and good, but that's really not what we're talking about. I think, I think it's interesting. I mean, look, I think we can start with AlphaFold. I think that's just fascinating, right? That we actually now have this enormous advance that we can actually predict protein folding. And now you're seeing, I think I just saw a paper, people are looking at, someone created a sort of a follow-on sort of software or AI application where they can look at peptide HLA interactions, right? And look at now that sequence as well as the biochemistry around that and the charges, et cetera, and they can now predict antigenicity and immunogenicity. So that's really exciting. So I think what we're going to see, particularly on the drug discovery, target identification space, and my colleagues might be even more adept, have other ideas around this, where there will be a greater understanding about how we can actually think about understanding biology, target validation, et cetera, in much more complex ways. I think it's going to take, actually, we're talking about biochemistry, biotechnology. I think there's going to be many, many more companies now that are spawned around specific applications of AI technology and how it applies to drug discovery and drug development. And I think that's where, so I think there's a lot that will happen. I just can't predict what it is today.
George?
Well, if anybody's been in this business, or almost any business, as long as I have, the one lesson that you learn is short the hype, okay? We use a lot of AI. We believe in information. We believe that it's very useful to process a lot of things, to yield better organized information. A lot of times you have to start with well-structured information. But there's no I. I mean, this is the biggest misnomer on the planet. There's no real I. I think of intelligence to me is what I'm talking about, innovation and creativity. There's no I in AI. We need a whole new kind of AI to come up with a great idea. None of the game-breaking technologies that I talked about could ever be in any way, shape, or form conceived by AI, because it can't conceive of a new thing. It's basically trained on everything we already know, and all it can do is reorganize it or reformat and so forth. I think in healthcare, in the industry, I think it's healthcare burden is one of the most important things for us individually as human beings. Nothing is worse than when you find out somebody that you care about has a devastating disease. Nothing is worse than that. But as a society, it's an existential threat. You know, 1 in 3 Americans, 100 million Americans are obese. You know, a third of them have type 2 diabetes. People want free health care. We have too many sick people to be able to afford it. It's going to crush us. It's just like climate change. These are two of the biggest existential threats, and what we need is we need innovation in these spaces. In terms of the healthcare problem, I divide it into 2 buckets. One is using the available tools to actually better manage healthcare. That is, right now, the average time to diagnose a complicated disease is measured in like 5 to 10 years. You know, you have one of these rare diseases, they can't figure out what you have. I mean, human beings, I'm sorry, we're not so good at that. I think the first place that we can use AI and machine learning is in that aspect of the healthcare burden, which we are in a sorry state as a country doing, but projects that many of us have been involved in, like the UK Biobank, those are great examples of how we can structure information, use that information, and actually use that information to much more efficiently deliver healthcare and not require, you know, mega brain physicians. I mean, the only reason it's so hard to get into medical school is you need a big brain to try to deal with all this unstructured information and make a diagnosis and a treatment plan. Machines should be much better than humans at that. That's what we should be striving to use these machines for, to actually revolutionize how we manage the healthcare burden using existing tools. In terms of the second part of the problem, which is we need new tools in healthcare. Most diseases, even in cancer that is so easy to measure, we are, I'm sorry, great advances, we are still so pitiful. You get a lot of diagnosis, you're going to die, okay? And so we need new technologies, new innovations. For that, AI will be a tool, but it's not going to replace what I've seen, like I described. There's big things, game-changing technologies that revolutionize things. Genetic medicines, I share the hope that those are going to take us all to the next level, but that's never going to come from AI. That's still coming from humans, unless a human being, just like they can design a Boeing, okay, they design a new form of AI that can be as creative as human. But we are so far away from that, and that technology would be entirely unrelated to current AI technology, which is just based on training of all known information and spitting it back out to you with a few variations.
Yeah, I think AI is really important for the industry in terms of clinical trial design, access to patients, the manufacturing pipeline, making sure we're not just wasting product by sending it to places where the patients aren't. There's a lot of uses for AI. Also, you know, really specifically within the drug development space, you know, there's a design space with small molecules that you can generate new molecules around that perhaps the really talented chemist wouldn't come up with. You then have to have a human to come in and, you know, make that and iterate and build that back into the system. And new small molecules are coming out of this. And this will happen with the protein design space as well. I think, you know, Pushkal mentioned AlphaFold, and, you know, there's protein design space beyond what we could even imagine, because we kind of assume two proteins do this, but they may do this, they may do this on a cell. It's very, very unknown. I think the one thing that I see will still be a struggle, and this gets back to that, you know, multi-parameter drugs, is will AI ever be able to impute the next generation of, say, protein degraders or molecular glues and say your target needs to bind with that target in this cell type under that condition within a specific tissue? I don't see that happening for a while.
I was just at a seminar yesterday in which somebody was designing degraders based on protein-protein interactions using AlphaFold and some thermodynamic stability measurements. Won't take so long.
No, I think agnostic. No, that's happening in the protein design space for sure.
I totally agree with what's been said. It will be an accelerator, and acceleration is a very important thing. But I also want to make one other point. Our major problem in this country, a major problem, is healthcare delivery. And we will not be able to make healthcare work for more people unless we have more people interacting with them. And I hope that AI and machine learning and support will allow us to have a cadre of people. We now call them nurse practitioners, or we can give them another name, that will populate more contact with communities and people in different communities and bring healthcare with them with the support of a system that has more information at their fingertips. If we have to train people for 16 years to put them in a medical office, we're not going to be able to provide the healthcare we can with the technology.
I totally agree, but we are in a sorry state of getting to that point. If we don't do that, I think we're going to get crushed by the healthcare burden. But we are very
That's-
far away from figuring out how to do exactly what you said.
Okay. An interesting new, okay, we've talked about genes and we talked about the new modalities to deal with them. We've made major success there. But really permanent treatments, one and done, is something that is becoming more accepted with gene therapy and gene editing and other technologies. And they're going to get better all the time. So if you look down in the future, will the sort of 8 to 10% of the people who are born with a genetic disease gene be treated with gene therapy and one and done versus other therapies that require continued treatment, annual treatment? How do you view the future of gene-oriented therapies in the current biotech pantheon of treatments, and what will be its impact? 10 years. What will be its impact in 10 years?
Well, unfortunately, related to what Ray was saying before, it'll be a lot less than people are hoping it'll be. We are very big believers in all of these genetic medicines. We have a great collaboration in Alnylam and sRNA, but we also have a great collaboration with Intelia on CRISPR-based approaches. We're in the midst of the first large phase 3 trial in the history of CRISPR. It's really worked to seemingly as a one-and-done treatment for this amyloidosis TTR-related disease, which also can be treated with siRNAs. But we're now midst of a clinical trial. The results of just that clinical trial are probably years away. And that's the first big example. We don't understand the true safety impact in the long term, whether you want to use this for simple problems like cholesterol lowering, altering your genome with some unknown risks. We're a long way away. It's very exciting, but it's going to take a lot of work and a lot of effort to really get to that point. But the one thing about all these technologies, and Pushkal and I were just talking about this because we're dealing with this, okay, people think it's the gene medicine that's the hard part. As Ray was getting at, there's actually a lot of ways to edit a gene if you wanted to, okay? The problem is right now, other than the liver, okay, you can't edit genes easily unless you take cells out and put them back in, which really doesn't benefit you hardly at all if you see what these sickle cell therapies are doing, because therapy will actually kill you because it requires a stem cell transplant. So what we have to understand, getting back to my point that I made earlier on, okay, you always got to try to think about what's limiting. And it's always innovation. You got to figure out what do you got to innovate around? What are you trying to do? Is gene editing, should you form a company and the total focus of the company is gene editing? Is that really the hard thing? No, it's delivery to the right tissue, the right cells in the body that you want. And, Pushkal and I, we're collaborating on working on this. We're working on this with Intelia. That's going to require, once again, we're all hoping things like antibodies, targeting, and so forth. I see Mark there. He was a pioneer in some of these approaches. We're hoping things like this might solve some of these delivery problems, but it's taking a lot of work. And that's, once again, getting back, to what the mantra that I was saying before. This business, it takes so much time because developing the technologies, perfecting them, getting them to work in humans, testing them in humans, these take years. This is why it takes so long. And we all see the promise. We see the hope. We see the possibility of AI There'll be accelerants, but nothing is going to really get us as fast as where we need and want to be going. And that's why this is the toughest business in the world.
Jane?
I agree that 10 years is probably too short of a time frame in order to see the changes. Yeah, I'm somewhat of a skeptic on some of the approaches that are being taken. I think George touched on it briefly. You know, do we really need genetic modification to control cholesterol? There's another example, it's an Intellia program in hereditary angioedema, where there's a monoclonal antibody on the market, there's an siRNA that's under development. You know, personally, if I think of it, you know, of me taking the medicine, would I want to gene edit my liver, all of the cells in my liver potentially, that's like what, 10 to the 12th cells, and take that risk of having an event in one of those cells for which a positive selection enables that cell to start proliferating, when there's a more conventional medicine that's available? And, you know, personally, I wouldn't want to take that risk. So to me, the focus, if I were working in that space, where Amgen's not working in that space, but if we were, I would be very insistent on going after a disease where there's really no other way to treat the disease, right? You're adding back a gene product, for example, that's missing, that you can't add back any other way.
Yeah, I think, you know, the field is certainly moving in this direction, and there are some great examples of where gene therapy works. We have our own, if you argue that ASOs are gene therapies, they're certainly, you know, correcting for providing the material for when you have a mutation, for example, SOD1 in ALS in one of our products. One thing that I really think about a lot, though, too, is sometimes when you have a really a disease that's driven by a genetic mutation like sickle cell disease, it's pretty horrendous, but not in everyone. And even in some diseases in ALS, you get sporadic versions of it, you get genetic drivers, but you can have a genetic driver and not actually develop the disease. And so at what point, just, you know, would you want to go into a newborn, say, and say, you've got this gene, you should take a therapy prophylactically, when we really don't know how that's going to manifest? And I think as a field, we just don't really know how to think about and handle this right now.
Yeah, I was just going to, you know, build on a little bit of what you said, Ray. I think, you know, I think there are areas where there will probably be a role for gene editing, but, you know, as we're starting to develop, again, ASOs, SIs that can be given once every 6 months, once every 12 months, working at the transcript level, reversible, you know, it's harder for me to see in certain applications where, you know, I guess I don't know that I would prefer the gene editing approach either at the DNA level, for all the reasons that Ray was saying. But I'm sure there are applications that make a lot of sense where there aren't other available therapies, and that, you know, I think that's where we need to focus. I think the other thing is just until we can kind of get to figure out, again, where George was talking about barriers, you know, how we get safer, better tolerated myeloablative regimens and things for patients. I think that's going to be a huge, huge barrier for, you know, for these types of therapies. And so it's another area where we really need to be advancing the field.
So there's a whole host of patients who are one patient diseases, one patient mutations. And they are extraordinarily damaging in many cases to a patient life. If we can even ameliorate some of that with gene therapy, it'll be a gift to a lot of patients. So I'm much more, I think there's a space for this, and it may ultimately be a very broad space, but I do think it's something that will be a big augmentation of what healthcare delivery could be if we are able to get it in a state of delivery that's effective.
One thing I think is fascinating, Phil, is this was just published in BioCentury, I don't know, a month or two ago. They were looking at spinal muscular atrophy, where you have 3 products. You have an siRNA, you have a small molecule, and you have a gene therapy. And it's interesting that the gene therapy is actually the poorest selling of the three products in that space, which almost seems in a way counterintuitive. You would think maybe that would have been the more obvious go-to, but it's an effective, successful product, but it's actually underperforming the ASO and the small molecule.
I think, but it comes down to what you say. It's a lot of it is the safety problem, and the fact that most of it is actually going to the liver. And so these are the things. If we solve the delivery problem, and I agree, this safety is always going to be an issue with a lot of these new therapies. But, you know, to Phil's point, I do think, and I think you mentioned it, that, you know, one major program or set of programs that we have with Intellia is repair and replace, okay, which can't be done with other approaches. And we know that a lot of these people who are getting replacement therapies, in some cases, they don't work because they have to be inside of a cell. And so you've got to figure out how to deliver a product that can't get into a cell in other ways. And so I think those are the sort of things that you guys are talking about where we will tolerate the safety risk. I also think TTR amyloidosis in people whose average age is over 80, you know, that's why we're in it with, you know, Intellia and that. So there are the settings where we have to pass the hurdles, we have to see how they're going to work, we go to the highest unmet need where we're willing to tolerate some safety risk, and then we'll learn, and we'll learn if there is a safety risk, can we then overcome it? But this is why this business is so hard. Even if you cure something, you might end up having an intolerable side effect. I mean, it's just a hard business. It takes time to figure it all out.
So we have time for a couple of questions. Well, your hand jumped up first.
I'm sorry. My name is Nellie Wild. I work for Perspective, and we were honored to be nominated for one of the digital health solutions, but that's not why I'm talking. Actually trying to avoid liver stuff, but you guys keep talking about the liver, and then I'm like, oh, I don't want to drink wine tonight. But because FGF21, are you familiar with that? Yeah. Okay, so my husband co-founded Akero Therapeutics with Tim Rolfe. And the ride behind that and getting the molecule and everything was insane, especially, you know, when my husband couldn't make payroll for all, you know, a few employees at the end of 1 month. But he got the molecule. He's a PhD, not traditional for this, PhD historian, lawyer, because you have to earn money in DC, but he doesn't like that. I just like wild rides. I'm silly. We had small kids, but it was fun. And watching him go through that was fascinating. But the other thing, too, is just having an advocate inside of Amgen. I don't know if you know the story at all, but there are other people, you know, breaking NDAs, you know, investors trying to get the molecule around Jonathan. Long and the short of it all is, I'm still jealous to this day of the person who found the Fabergé egg, you know, at a garage sale. But, like, is this standard in corporations? I mean, I know the therapeutic was about ready to expire. I mean, it's crazy stories about shipping it in time and all that. But do you have things for rare diseases, or because programs have been discontinued? Like, and how would somebody, you know, access them? Or is this normal, like, to have people hear about things and all that? Because that's how it came about with us, word of mouth.
You know, that's a very interesting example. The FGF21, efruxifermin, I believe is the name of the molecule. It was an Amgen molecule. It's for metabolic-associated steatohepatitis. And, you know, that was a program where scientifically it's a very interesting area. It's a very challenging area. There's a huge population in the United States that is on the pathway to having MASH and eventually cirrhosis in the millions. And we understand the genetic underpinnings of at least some of the cases of that disease. And, you know, until earlier this year when resmetirom was approved, there was literally no approved drug on the market. And so, you know, I'm getting back to a comment I made earlier about, you know, consistency of support in a large company where you have people turning over at a certain rate. And also, you know, what is sometimes a conflict within a large company between the research side of the company and the commercial side of the company. And the whole discussion around, and there was an issue, a scientific medical issue with MASH, in that the FDA was requiring liver biopsy, which is a real barrier for measuring the progression of the disease. And so considering that barrier of having to use biopsy as the measure, this other conceptual barrier of what's that market going to look like when a drug exists, because there's no existing drug? Are you going to be able to identify patients? Are you going to be able to get payers to pay for those patients? So this program ended up on the shoals, despite the excitement and research over this program. Yeah. And so, I would imagine this is not super rare. Amgen is not the only company where this has happened, where you have something that's scientifically interesting, but then you have to say, you have to get the rest of the company to believe that you can pull this into a product, and that product will make a meaningful difference in people's lives, and payers will reimburse for it, doctors will prescribe it, right? And this was the challenge. I think now a lot of that has changed in the past few years, but when this deal was done, the environment was very different.
I think too, just to add to this, and it gets back to the original question around, innovation in the ecosystems that we have, and more industry partners, or really has relationships with these patient advocacy groups as well. And so there's an example of that N of 1 patient. It's not even ultra rare; it's nano rare, right? Alnylam, that will really partner with industry, and in that example, it's using ASO for ultra or nano rare diseases, and really working with partners like Pushkal, myself, and Biogen, Ionis, other companies that have worked in this space, to understand their technology, to understand the therapeutic index, what's the kind of potency you need in a molecule, share that knowledge, and then help fund them to go off and actually work with the FDA. And then they've established these relationships with the FDA so that they can think about fast tracking, like the whole asset gets some kind of approval, and then the widget, the gene that you're correcting, is kind of the plug and play that you could think about.
We have a question.
Thank you.
It's got to be short, and the answer's got to be short.
I'll do my best. Okay. Kayvon Jared Pfizer, thank you for the insightful discussion. I wanted to connect two really important points that have been made. Innovation and huge healthcare burden, and add another factor in incentivization. Innovation doesn't occur in a vacuum. It's driven by incentives and driven in a certain direction by incentives. So we have plenty of examples as treatment as prevention, but usually there's cultural acceptance and a lot of incentives for treatment when a disease has already happened. And even when it's secondary prevention, dyslipidemia, obesity, we have to label it as a disease first, even though it's actually trying to do some secondary prevention. Are there commercial incentives, or are the business models going to be able to accommodate more primary prevention as treatment, sort of this vaccine approach that you said, sort of these one and done approaches? Do we have models in place that can accommodate that with respect to incentives?
I would say no. This is what I was saying at the beginning, that we need a lot more incentives. Ray and I were just talking about a class of drugs, which also Alnylam got involved in, using an siRNA approach, that I think almost every American should be on in terms of prevention, which are these PCSK9s. For most of us, unfortunately in America, due to obesity and other lifestyle issues, cholesterol levels are so high that what you're on statins is still not enough. Statins help you, but they're not getting you into what which should be the sort of pre-industrial range. I think like I said, almost every American, they're so safe, should be on these PCSK9 blockers, whether they're antibodies or siRNAs or whatever. Okay, the incentives do not exist, and in fact, the system destroyed the class. A lot of people, Chris walked out, but when Chris and I were involved in developing these, we thought these were going to be the biggest drugs that everybody would be on, and I'm sure you guys were thinking the same thing. And very few people are actually on them. So this is what I'm talking about at the beginning when I'm talking about the most important. I want to parrot what you said. The most important thing is innovation if we're going to treat healthcare burden in a new way. Okay? We can do much better with healthcare delivery. That's one issue, but we don't have enough tools. When we create tools, the incentives aren't there to, A, be able to give them to enough people. We don't want to give them out for free because you know how much it costs us? It costs over $2 billion to develop our PCSK9 blocker. We're never going to make our money back. I mean, you know, in terms of return on investment. So this is what we have to understand. And we do a terrible job of communicating this, as I was trying to explain before, how important this is, how critical it is. And if everybody just all wants this for free, okay, where are the incentives going to come from? So I haven't thought of a way to fix it, but incentives are the most important thing, I believe, to drive the right kinds of behaviors, and we don't have enough of the right incentives. And this is just one example of a great prevention treatment approach that almost everybody should be on, and almost nobody is on because we don't have the right systems in place.
The time has passed. I want to thank the audience. It's been wonderful having you. I want to thank the panelists. I've enjoyed the conversation enormously. And I wish all of you a wonderful afternoon.